Joseph Buxbaum, thank you so much for joining us today. Take it away.
Thank you for the invite, and I apologize in advance for this complicated talk. I'm basically gonna what I'd like to do is kind of give you an update on where we are with the genetics of autism, and then talk about how that might kind of lead to precision medicine, and talk a little bit about what we're doing at the Seaver Autism Center in that regard. I do have some disclosures, but I don't think they're directly impacting anything I'll talk about today, but they're down here at the bottom. And let me try advancing here. So this is a slide which I showed to a bunch of people in biotech a couple of years ago about this new syndrome called Miseryoff syndrome. NDD stands for Neurodevelopmental Disorders. And I said that there's this new syndrome where the cause is unambiguous. There's an easy and accurate test. There's very high conversion, by which I meant that if the test was positive, you would have the syndrome. There are cell and animal models with high construct validity. There are translational biomarkers. There's preclinical evidence that reversing the causal deficit will lead to improvements. The prevalence is kind of in the orphan designation range, but not terribly rare. There are family foundations that are very engaged. There are registries of patients. There are expert sites doing extensive phenotyping, and they can identify clinical endpoints. The FDA is very supportive. And there's probably a way to get, if the drug works in this particular syndrome, then it probably could be tested in other related syndromes. And I put this up and I said, if you knew, at the end, when I tell you the name of the syndrome, do you think it would be a target for drug discovery and development in biotech and pharma? And everybody said absolutely yes. So we'll come back to that in a moment. So when you think about genetics, can you see my pointer?
Yes, we can.
Okay. So one of the ways people think about genetic variants is how frequent is the variant in the population? So that's allele frequency down here. And what is the effect size? If you carry that alternate allele and you're looking at some phenotype, does it have a tiny effect or does it have a major effect, right? So for example, a Mendelian disorder is where, these things that are very, very rare, but if you have that mutation, then, there's an effect, there could be 100% chance of getting the disorder, right? And for obvious reasons, although I'll explain them, things down here that are both very rare and have tiny effect sizes, we're not going to find. We already know that for some things, even if we sampled every human being in the population, we won't find those things. And then the other area we can kind of ignore for autism and related disorders is that if something is both common and has major effect, okay, it's probably not going to, it's going to be subject to purifying selection. You're not going to find that common variant of major effect in the population, because when like just to put it clearly, autism is associated with a very, very reduced fecundity, as are many psychiatric disorders. And so it's not possible that there could be something both common and have major effect on a neurodevelopmental condition. I mean, you probably have heard of ApoE4. ApoE4 is right up here with the allele frequency of something 20 to 30%. And the effect sizes are probably between 3 and 10 for fold increased risks of Alzheimer's disease. But Alzheimer's disease happens after reproductive age. That's why we can have a common variant of major effect in Alzheimer's disease. So what we're looking for in autism and all the developmental disorders are along these in this kind of path right here. And taking advantage of some of the very, very unique genetic architecture in Sweden, we were able to actually parse out and ask the question of what proportion of autism risk is these common variants. These are the variants on the bottom right, genes of variants of tiny effect, and how much are kind of more rare variants of major effect. And then we're also able to estimate what used to be called environment. We have to be a little bit careful because people sometimes confuse environment with what's in their plastic water bottle, but really it's just unaccounted for. It's a term called the E term, and it includes many things that aren't formally strictly environment. So we said unaccounted. But what you can see is most of the risk for autism comes from those SNPs or those common variants of tiny effect, and very little comes from things that are have much more significant effect. However, a variant here may account for much of the risk in an individual, while this common variation may account for much of the risk in a population. So starting with common variants, I'm only going to talk about them very briefly. So down here, these are things that have allele frequencies of 5% or higher. Their effect sizes are typically below 1.1. And this is the first kind of really, this is the first study in about a year ago now, where there were clearly GWAS, genome-wide association study hits in autism that were significant. And as I said, so there's, we think there's about 5, this is also including other data, let's say there's about 5 across autism. But importantly, the effect size for these loci is very, very small. And this is kind of what you see in schizophrenia, for example. If you've seen the very famous plot in schizophrenia, you see all these peaks, but each peak increases risk by a relatively small amount. So the other part of this curve, or this diagram, are these rare variants with very high effect sizes. And there, in autism and intellectual disability and epilepsy, there have been, and other things, there have been major advances in the past few years. And one of the groups that's really pushed this forward is the Autism Sequencing Consortium, which basically took advantage of the fact that there were many sites all over the world that A, wanted to collaborate, and B, had large samples that had not been sequenced. And so, in 2010, we kind of started this whole thing, and we began to kind of reach out to people that we knew through other studies, particularly through GWAS studies, and say, would you like to participate in the ASC, and we will figure out how to get your samples sequenced. So you can see samples from Sweden, from Mount Sinai, from Germany, from Japan. Like over time, we've gotten more and more samples from different parts of the world. And we have a few papers, but the most recent one just came out early this year, and what we call ASC2, because it's our second major data freeze. And these are some of the people who are very active in the working group, although many, many, many people were involved over the course of many years. So this is the paper, and I'll go through it kind of just to highlight some of the interesting things. So first of all, there are 102 genes that we would say we have found so far that are highly associated with risk for autism. And there are various thresholds here. This one is basically Bonferroni correction. This is FDR correction at a 0.5 or 0.1. So the further higher up you go, or the lower the p-value, the greater the evidence. Everybody would accept that these 26 genes are genome-wide significant. I consider FDR 0.05 also genome-wide considered significant. So we have about 100 genes that kind of we now can say are significantly associated with autism. And the important thing to remember is, unlike those Manhattan plots I just showed you, this is actually mutations in the gene. It's not a SNP that's in the gene neighborhood. It's actually an amino acid change in the gene. And at least for most, certainly for everything, these top 20 some odd, the effect sizes are very high. In other words, it's 20-fold increase in risk on average, if not more. So I'm going to kind of go through some of the, with all this data, this is, in case you're wondering, 10,000 cases and about 3 times as many controls, either as parents and siblings or in a case control design. So 30,000 plus samples. And as was already known from previous work from the ASC and others, many of the genes that were coming up are genes that are involved in regulating the expression of other genes or chromatin remodeling. And then, not surprisingly, there are a significant number of genes that are involved in what we call neuronal communication. These are genes that, in neuroscience you're quite aware of. We began to see for the first time significant emergence of cytoskeletal genes in this big study. But the main two categories are what we call GER and NC. Excuse me. All right, so, when we these classes, the purple and the green, we knew about for quite some years. And it was kind of easy to say that maybe these genes are regulating these genes, and that's the mechanism of action. So with this large data set, we're now able to formally test that. So everything in purple here are the genes that are involved in regulating other genes. And what's in green here are genes that are synaptic genes, and what's in gray are the cytoskeletal or other genes. All the genes here have significant, like we know many of their targets, okay? Sorry about that. My mouse seems to jump forward. So we could ask the question, for all these genes with known targets, what is the evidence that they are regulating any of the genes down here? And the answer is none. So it is not the case that the GER genes are having their impact by regulating synaptic genes. These GER genes have no targets, but you can see that here, these clearly regulate these genes and have no effect on the cytoskeletal genes. So two major classes, functional classes of genes, and they don't coalesce on a single kind of pathway, if you will. Nowadays, with single cell data, you can also begin to map out cell types where you're having your major effects. So you can map all the autism genes to different times during development, right? Which is kind of going from top left to bottom right, and also in different kind of specific cells. And here we have the inhibitory and excitatory lineages, and here are some other non-neuronal cells. And what you can see is over time, but because this is time, it's quite clear that the autism genes are expressed in later development, kind of mid-fetal, late fetal, in both the inhibitory and excitatory lineages. So, it's neurons for the most part, but it's not just excitatory or inhibitory, right? Very little microglia, maybe something in astrocytes and nothing in endothelial cells. So that kind of highlights again and again, I think, the complexity of autism. There's also some real kind of jewels that came out of these big studies. For any gene with multiple mutations that's significant, we can figure out whether most of the mutations are loss of function protein truncating variants, PTVs, or missense variants. And what you can see is some genes are always loss of function, and some genes are almost always or always missense. And that has begun to, and when you see genes like this that are almost exclusively missense, it tends to speak to a gain of function or a dominant negative effect. So DEF1 is one of the genes, and what you can see is everything we see in autism, everything we see in other developmental disorders is a missense mutation, and it seems to be acting through a dominant negative mechanism. The protein is expressed, but it cannot dimerize or bind DNA, so it's competing for, it's inhibiting other wild type proteins, basically. I'm sure you know that there's lots of evidence for copy number variants in the human genome, and very strongly associated with ASD and other developmental disorders. So these are deletions and duplications in the genome that increase risk for various disorders. Some of them are recurrent. They happen over and over again because of specific characteristics of the genomic sequence around them. And many of them are quite large, 16p11, 22q11. And it's been very hard in many cases to map things to a specific gene or one or two genes. So since we have so much sequencing data, we are now beginning to say that within, for example, 2q37, the primary gene that contributes to phenotype in that deletion is this gene HDLBP. While in this interval over here, SHANK2 was known, but we show that KMT5B is also contributing to risk in that same interval. So we're able to get from these big gene copy number variants to specific genes in some cases. So the next thing we did, because people have always been concerned about specificity, it's been very well known that, by the way, if you want to interrupt, please do. I'm not looking at all your faces, so I can't tell if I've lost everybody or only 50% of you. So, don't be shy. It was already known as we found more and more autism genes that they overlapped very significantly with genes for intellectual disability and global developmental delay. So if you remember from my slide, I had this ADMP gene, SYNGAP was one of the autism genes, SNC2A was a major autism gene. It just goes on and on. This, however, plot is for mutations in the so-called DDD, or Deciphering Developmental Disorders study, which took place in the UK, serially ascertained about 12,000 individuals with developmental delay syndromes and sequenced them all as trios. And so there's enormous overlap in genes, and so people began to ask, is what we're finding in autism and or ID, intellectual disability, is there any specificity whatsoever? With the sample sizes being so big, we're able to actually answer that question. So the most important thing we did was we first did a heterogeneity test. We asked whether, when you can take all those genes together from intellectual disability, developmental delay, and autism, and you put them, and you ask whether they are similar in various parameters, they're clearly not. There are clearly differences between ID genes and ASD genes, even if there's lots of overlap. So once we demonstrated there was very significant heterogeneity, we just made a Bonnet binary classifier, which is of course not perfect. We did a split and we said genes that are in orange, more mutated in the autism cohort are down here. Genes that are more mutated in the DDD cohort are up here. And we can ask now, are there any kind of general differences between people carrying these kinds of mutations versus these kinds of mutations, right? So I'm not pretending to anybody that this gene is an intellectual disability gene and this gene is an autism gene. Only a few genes are so far away from the midline here that they're actually significant. But we've proven heterogeneity. We've shown that the genes from the two different, the kinds of mutations and the kinds of genes from the two different data sets are significantly different. So we're doing just a classic, classifier binary split and asking, what do we see with all these genes clumped together? And so if you go back to your autism sample, and only the autism sample, because that's all we looked at here, and you ask, is there any difference between those individuals that contain the ASD predominant genes versus those that carry mutations in the ASD and NDD, neurodevelopmental disorder genes, the more broad genes, or those individuals in the cohort that have no variants in any of our genes so far? And what you can see is age of walking is much more delayed if the individual is carrying a gene mutation that's associated both with autism and intellectual disability developmental delay. While if it's the so-called ASD predominant gene, there is a significant left shift of age of walking. So there's a significant motor delay with the genes that have a more global impact. Full-scale IQ, same thing. The genes that have the greater impact, in the blue line here, have a lower full-scale IQ compared to individuals carrying mutations in those genes, have a lower full-scale IQ than individuals carrying mutations in the ASD predominant genes. And they, in turn, have a lower IQ than those individuals in our cohort that we didn't find any of these mutations in. So this very crude classifier it really kind of predicts kind of more severe phenotypes, if you will. And interestingly, and I won't have you read all of this, these less severe genes, the ASD predominant genes, are much more likely to be inherited. So the NDD genes being so deleterious, if you will, it's very unlikely for mom or dad to carry the mutation and pass it on. While for the ASD predominant genes, because the impact is smaller, it can be transmitted. I should say that much of what we're finding is de novo. I probably should have said that earlier, which means that it's a mutation that occurs in the egg or the sperm and is not carried by mom or dad. Okay, so that's where we are with the study so far. We keep on pushing forward. The Simons Foundation has created this very large database called SPARC. currently 27,000 individuals. They want to get up to 15,000.
Sorry, I have a question because
Yeah.
the NDD genes was more heredity, but when you did the classifier, your genes, was based on de novo or heredit mutations?
So we classified them based on, that's a good question. I think we just use all of our genes and all of their genes rather than sub-select our genes for de novo only. I'd have to check that.
Okay.
Yeah, Joe, I also have a question.
Yeah.
Rebecca, did you stratify by intellectual disability in the association analyses?
No.
And what was there a threshold for inclusion?
Nope. So the ASD takes all comers. I took out a slide just before to show you that we can show very clearly that irrespective of IQ, there are genes to be found, more genes to be found in those with higher IQ, lower IQ, you name it. So we did not pre-screen by IQ, and that's why it's appropriate for us to look at IQ as a function of type of gene.
Okay, so as a post hoc analysis. Yeah.
Joe, this is Alison. I have a question too, actually, about the ASD where the variants are inherited. Do you see phenotypes in the parents as well, and what kind of phenotypes seem to be associated with the variant in the parent?
So the answer is a little bit more complicated. You know, most of our power, right? Most of our power actually comes from the de novo findings, right? Okay. Because de novo findings are so rare, and we actually have mutation rates for each gene in the genome, it's very easy, you know, three de novo mutations in a single gene are pretty much, unless it's a titan, are pretty much sufficient to make it a significant finding, right? We are getting very little power from inherited genes because the signal to noise is terrible. We get lots of our power from de novo mutations, and we get a significant amount of power from case control data, but the case control data includes de novos that we just don't know are de novo. So most of what we're finding, most of these findings are driven by de novo mutations, but there are examples, both in this study, but also in prior studies, where you see a mutation that's kind of inherited. And, you know, typically, there is some kind of impact on IQ, maybe mild intellectual disability, there may be autistic traits, there may be other psychiatric disorders.
Okay, thanks.
All right, so Spark, and I won't go into it, but we're still cleaning the data, but Spark will basically double our sample size. And so we expect that by the end of this year, we'll have a sample size that's over 50,000 samples, including, so these are so-called family-based or trios, and this is the case control data. This is from Denmark, and this is from Sweden. This is the work my group did, and this is the work that, well, the EyeSight Consortium in Denmark has collected. So I won't show you the data because it's still being cleaned, but not surprisingly, there are more genes. So if you kind of look at the ASC and other major efforts over time, you know, our first study with just a couple of hundred families, you know, this is the number of cases we had. We found 9 genes when you put all of this together. This is the Derubi's paper. So Sylvia Derubi's was first author on this. This is the, I apologize that I keep on jumping. Something about my mouse. This is a paper from 2014, had about 4,000 cases, had about, you know, well, 60-ish genes. Then Stefan Sanders and others put together the ASC data and the Simon Simplex data, the historic SSC data, plus CNVs, sample size a little bit larger. This is the number of genes we found here. This is the paper that just came out, 10,000 cases. Here we are. And then this is with Spark. So this is everything here, everything before this, plus Spark, 20,000 cases, and we anticipate, we already know we already get about 185 FDR corrected genes, and just a small increase in FWER corrected genes. So, and it's still pretty linear, right? And we've known this for years, that we've got a ways to go. Okay, so we're going to keep on building. We have more samples I won't go into. You've heard me say that we're underpowered for inherited variation, but we're just now getting to the point where we're getting some power on that. And so a major focus when we have, especially when we have the Spark data, will be to target inherited variation. We'd like to integrate rare and common variation and make a combined kind of risk score. Chromosome X is very interesting. Chromosome X, for the same reasons that we have underpowered here, we're underpowered here. Remember that most people with a chromosome X mutation are getting it from their mothers. And mothers don't have that many de novo mutations, so most of it's inherited. So mom who has 2 copies of X may not have a phenotype and will pass it on to a boy who will have a phenotype, but it is inherited, right? So we're underpowered on X, but we have some ways, including the case control data and also the much larger sample sizes, to begin to actually make findings on chromosome X. I should say that the DDD just published their chromosome X study, and one of the things that they showed, which we were all beginning to suspect, is that traditional genetic approaches on X, where you collect a family that has multiple males affected, and you see kind of, looks like X-linked inheritance, have probably identified many of the genes on X already. So there may not be that much more to be found, but at least with genes of major effect, but it's something we are very actively pursuing. And then of course, functional dissection is something that the ASC is trying to do, both on a lab by lab basis, but also possibly on a large scale basis. Oh my. Okay, so that's kind of, that's my kind of take on the state of the art of autism genetics. And I want to switch to precision medicine. So in precision medicine, this is kind of a derivation of a slide that Mark Baer shows. If you have a disorder and you can find some etiological component, either a gene or an environmental factor, you're able to model it in either animals or stem cells. And then you can study the brain, look at pathophysiology, think about drug development, think about novel therapeutics, and then put them back in patients, right? So that's kind of the circle. And we've kind of committed to this circle quite some years ago, more than 10 years ago. And we kind of think that we're able, as a center, to provide kind of 4 levels of in that circle. First of all, we are doing a good job at knowing the targets for precision medicine. We and others are able to make, obviously human neurons in vitro. We and others are able to make mouse models. And we and others are able to look at patients that carry those rare mutations and ask questions about their phenotypes and possibly develop clinical endpoints, right? There are some things we don't do well, at least not in the Seaver Center. One is high throughput screening, which would be here. And the second would be MedChem, which would be here. But obviously within Mount Sinai, there's expertise, and there's also, presumably, biotech and pharma might be interested in collaborating at the right point. Okay, so just to give you a quick overview, we study 4 genes on all those levels of analysis: SHANK3, ADMP, FOXP1, and DDX3X. DDX3X is on the X chromosome, which is why you don't see it here. These are the 3 that we study, plus 1 you can't see, DDX3X. What you can, and I'll talk primarily about SHANK3, but DDX3X is probably 1 to 3% of intellectual disability in girls. FOXP1 is about 1% of autism. ADMP is about 1% of autism. SHANK3 is about 1% of autism and ID. So all of these are rare causes of autism or intellectual disability, but they're not that rare, right? Fragile X is in the same order. Rett syndrome is actually much rarer than these. Rett syndrome is maybe 0.5% of intellectual disability in girls. And here we have a new syndrome that nobody's heard about that is 1 to 3% of genetic ID in girls, right? So these syndromes, even though they're rare, they're as common as things you've already heard about, like Fragile X syndrome or Rett syndrome. So that's why there probably is a path towards precision medicine and kind of orphan designation with these kinds of genes. And all these genes are being studied by somebody in this context. DYRK1A certainly, SYNGAP, SNC2A, many people, CHD8, and so on, ARID1B. So it's not just us. And just to remind you that whoops, wrong place. To remind you that all of these genes we're talking about have very rare mutations of major effect, right? So if we knock out that gene in a mouse, we have high construct validity. All right, so we think we know a lot of the top genes, and we've chosen 4 of them.
Joe, can you clarify how you chose those 4 genes? Was it just historical coincidence or specific rationale for those 4 versus the others?
Opportunism, I think. We were sequencing SHANK3, and I'll explain why in a minute, even before it became an autism gene. And we were already making a knockout about that time. FOXP1-ADMP, we had a lot of interest in, but also the family foundations came to us and said, we'd like to support you doing this. They gave us seed funding to begin it. DDX3X, you know, this is the most significant cause of ID in girls. So as soon as we learned that, we jumped on it very quickly. As you know, autism in girls is still a not fully characterized situation, and so this gives us a kind of a window on autism, but it also gives us, you know, the most common genetic cause of ID in girls and developmental delay in girls. It's a pretty significant gene. We suspected that this was going to be a real gene, and we also were pretty clear that it was 1% of ASD already 10 years ago. That's why it came to the top. And these other 2, as I said, it was an interest. They're the top genes. But also the family foundations for each one of them came to us and said, we'd like you to study this. And everybody ranks their, you know, somebody picks this gene because they look at synapses. Somebody may take on this gene because, or and that's probably not a good example, but they'll take a gene where they, you know, they believe that that's an enzyme, so they can develop they can kind of do high throughput screens for enzyme, you know, for activators of the gene, for example, right? So everybody has a different reason. We had kind of more neurobiological / opportunistic drivers.
Thanks.
All right, so we have these molecular targets, and as I said, I'm going to focus on SHANK3, and I'll just say kind of, which partly answers Rebecca's point, even 50 years ago, we knew that at the very tail of chromosome 22, there was a gene associated with developmental delay, autistic traits, language delay, intellectual disabilities, seizures, and so on and so on. So why do we know that? Because, you know, when karyotyping was, you know, just a very, very low resolution, ring chromosomes that physically made a ring, were still detectable, right? And ring 22 is a relatively common ring chromosome manifestation. And basically the P and the Q arms of the chromosome end up fusing together, and genetic material is lost at the point of fusion. So we knew that at the very tip of either the P or Q arm of chromosome 22 or both, there was something going on that produced you know, feeding problems, delayed milestones, low IQ, verbal delay, anxiety, some kind of, I would even call it atypical kind of bipolar, if you will, and autistic traits, right? So 50 years. And then as the methodology got better and better and karyotyping got better and better, people began to map out the size of deletions either in ring 22 or in just kind of terminal deletions, and you know, smaller and smaller and smaller. And in the end, there was only 3 genes that here's SHANK3, 3 genes that seemed to be common to everybody that was being studied. And then the next step was, and this was in 2006, a recurrent breakpoint in SHANK3 was found in this syndrome. And so we and others were sequencing the gene pretty intensely. It is the most GC-rich gene in the genome, at least I think it is the most GC-rich coding gene. It did not sequence well. We never got some of the exons, and even in the published literature, some of the exons are missing still. But the other group, Thomas Bougeron and Durand and Betancourt, were sequencing as well. And this is what they found. They found 3 examples of SHANK3 mutations in a cohort ascertained for autism. First is the deletion. So that's already a well-known thing, subtelomeric deletion of chromosome 22. It's de novo, and it causes autism. This is a trisomy. So dad has a balanced chromosome translocation, no phenotype, but as a result of the, I said translocation. As a result of the translocation, he has, his kids could be monosomy for 22q, disomy, typical, or trisomy. And we have actually several families like this. So a person like this will have no phenotype, but their kids are very high risk for having monosomy of the region. And this individual has autism. This individual has Asperger developmental delay. So there's a gene dosage thing, which I'll come back to. Too little or too much for many of these genes cause a phenotype. And this is the single point mutation that they found, right? So this is a germline mosaic. It's a de novo mutation, relatively, I mean, it's germline mosaic. And both of the boys have this kind of insertion mutation. And it got published in Nature. Well, first it was a very major finding, but but also it was because of all that other data about the gene that even an N of 1 was sufficient to convince us that this is a real finding, right? So a big deletion causes it, a translocation causes it, a small deletion causes it, and guess what? Even a point mutation, a premature stop in the gene causes this the syndrome, which is now called Phelan-McDermott syndrome. All right. So obviously, again, if you know the gene and the gene is a major effect, you can do in vitro work, induce neurons, and so on. I won't go into this. We just got the proofs yesterday, so congratulations to Michael and Andy, and to Elodie and Kristen and Gabrielle and Sophia. We took 7 individuals with this syndrome and 6 of their sibling controls, and we differentiated neurons and NPCs, and we looked at gene expression, which is something that hadn't been done before. So it's 15 participant-derived stem cells, so it's a fairly big number for this business. All right, I will never use my mouse again. Bear with me. Best out of 3. Okay, so not touching the mouse now. In both NPCs, panel A, and induced neurons, panel B, Shank3 is down. That's a good thing, because these are heterozygous individuals. This is the volcano plot that Michael Breen and Andy did the analyses. First of all, if you look at panel C, there's good, reasonable correlation between expression and iPSCs versus NPCs when looking at fold change in cases versus SIBs. And then D, E, and F show you some of the pathways that are dysregulated. And I'd maybe just jump to G. Well, one thing, so the overexpressed in the iPSCs were more neuronal, while the underexpressed were in kind of development, including Wnt signaling. So what you can look, if you look at the middle of G, they're the Hox genes on the left, down in both NPCs and neurons, and then there's a significant number of Wnt genes that are down. Wnt signaling, regulation of Wnt, and Wnt binding. And I won't show you the slide, but then Michael went through the literature and looked at all other gene expression data that was out there from much of the gene expression data that was out there from other autism syndromes or studies, and Wnt seems to be a recurrent finding. Okay, so again, you know the gene, it's a high impact gene. If you knock it out in a mouse or a rat, you will have something that has construct validity. There have been probably 15 different rodent mutations made of Shank3. We've made 3 of them. So, and just to show you, if you look at, for example, I'm going to use my mouse. This is repetitive behavior self-grooming. Red is up. So in many of the models, there's significant evidence for increased activity, repetitive grooming. Blue is down. In many of the models, there's deficits in social behavior. There's certainly deficits in motor function in most of the models as well, and some possible learning and memory deficits. So each mutation is different parts of the gene. It turns out that there are 6 start sites in the gene. It's a complicated gene. I will say that we finally decided to make a mutation that knocked out all the starts, all the transcripts, because we originally made, whatever this is, we originally made one that just knocked out exons 4 to 9 because it was very easy. They're right next to each other. But then over time, we began to realize that there are multiple start sites, some downstream of exon 9. And so we finally just retargeted and knocked out exons 4 to 22. And the phenotype is definitely stronger. This is an early study I won't go into in great detail. This is a heterozygous animal with the early exon 4 to 9 knockout. This is LTP, which I think most of you know. The wild type LTP, the LTP in a heterozygote, missing just one copy. So it initiates normally, but it degrades more rapidly. For a variety of reasons, IGF-1 was of interest in the field. IGF-1 was already known to impact postsynaptic density, PSD95 expression. We knew that these mice had deficits in kind of the excitatory synapse. And so we gave them IGF-1 for a couple of weeks. And what you can see is the electrophysiology then goes back to normal. They have motor abnormalities, as I mentioned. Here's the wild type on a rotarod. Here's the heterozygote on a rotarod. Here's the heterozygote with IGF-1. More recently, Hala and team did a very elegant job on a rat model of Phelan-McDermid syndrome, so SHANK3 deleted in rats. And she noted that long-term social recognition memory. There you go, long-term. I was starting S with short. That didn't make sense. Long-term social recognition memory was impaired in the heterozygotes and the homozygotes. So basically, what long-term SRM is this: you put two animals together, and then you wait a day, and you put those two animals together with a new animal. Okay, and the nature of rodents, or at least rats, is that they'll spend more time with the novel animal. There's also short-term SRM, and the animals do perfectly fine in short-term SRM. So they remember things for a while, but they don't remember after a day. So for example, I'm not showing the wild type here, but if you look at how much time with saline the animals spend with the black rat and the gray rat, they're not black nor gray, but just for convenience, when you give them oxytocin, they now somehow remember who they met yesterday. Okay. Now, Hala didn't try oxytocin by accident. Oxytocin is known to be important in this long-term SRM, and so there was a clear deficit. As you can see, they had no record. The wild types look like this. I'm sorry, I'm not showing it. But the hets and the knockouts don't show any kind of preference for the new rat unless they've been given oxytocin. Then they kind of recollect that. The other thing that Hala showed was that they actually have deficits in attention. And lo and behold, the same oxytocin treatment actually improved attention in these animals. So this is the first time that you really see an effect of oxytocin on attention. Okay, and then just to jump to patients, Latha Soria, Alex Kolevzon, and Paige Seiper, and many others have been actually bringing in families to the Seaver Center with mutations in SHANK3. This is the first paper from 2013. I want to highlight, so 32 individuals, right? 75% meet strict criteria for autism, another 10% for the spectrum. And look down here in terms of IQ. 50% have profound ID, and another 23% have severe ID. So these are very impairing conditions. Sylvia, Paige, Alex, and others have then also gone out of their way to ascertain individuals with point mutations. So the paper I just showed you was all deletions. That's what we had easy access to a decade ago. Now more and more people have been sequenced, including people that we've sequenced and identified. And so this study looks at what is the phenotype when there's only a point mutation and no other genes are missing, and it looks very similar. It's less severe, but very similar. Okay, so remember I told you that IGF-1 worked in mice. One of the reasons we tried it is because it was being tried in Rett syndrome, but the other is because IGF-1 is very safe. It's used in kids all the time with primary IGF-1 deficiency. They could take it for 6 years. And so Alex got a waiver for an IND and took him 30 days to get approval from the FDA to do a trial. And In the social withdrawal / lethargy scale, there was an improvement on drug compared to placebo. In a very small, really a safety and preliminary efficacy study. The drug is extraordinarily expensive. The company that made it couldn't make it for 3 years. It's been a long, long haul. But Alex and together with Paige and also Michael Breen have actually done a replication. The thing that replicates, it looks like, is the repetitive behaviors, which were significant. I didn't show you this in the first study, nominally significant in the second study, but going in the same direction and significant overall. Still, only 10 more individuals because the drug is so expensive and hard to get. All right, and then, you know, guess what? Oxytocin is also a very safe drug. This is, you know, so Alex has a protocol right now trying oxytocin in SHANK3 patients or Phelan-McDermid syndrome patients. This is not oxytocin in autism, which has got a checkered history. This is oxytocin in a specific syndrome where there are changes in the oxytocinergic pathway, and Hala will tell you that actually the oxytocin neurons don't look so good. And then another clinical trial that Alex is doing, this is a company that has a compound, AMO is the name of the company, that it's an antiepileptic, and in preclinical models they show some efficacy. So now we have a trial going on for this their compound in the SHANK3 patients with epilepsy. Okay, so as I said also before, you know, we as a Seaver Center and possibly even as an academic medical center can do these things really well. With Paul Kenny and the DDI, we're able to do high throughput screening, and the same group can also do MedChem. But this really is, you know, for the Seaver Center, a significant gap. And so we created a new arm of the Seaver Center called the Seaver Drug Discovery and Development Program, and we hired Anna Kostic from industry to kind of help manage this. And she's been very successful over the course of a year. We have a formal signed relationship with a company that does QState, which does electrophysiological based high throughput assays. We have a formal relationship with Scripps that Paul Kenny helped put together. We have a all but signed agreement with Takeda, and so on and so forth. There's a few more in there too. So I think with Anna's help, we're finally, you know, getting the message out that, you know, there's something to be done here. Okay, so this is an opinion part of it, and I could skip it or I can go pretty quickly. Are there many more questions? I know we have 10 more minutes. If people have questions, now is a good time.
Yeah, so I have one, Joe.
Yeah.
Looking at, so if you, with the information you have from all of the sequencing analyses about specific genes, how many of those genes map to the loci that were identified in the GWAS? So do you expect that, is that right? Wow. So,
but remember the GWAS only found 5 loci, and it was significantly underpowered.
So if you set the threshold lower, so you're not just looking at GWAS significant loci, but, you know, I don't know, 10 to the minus 5 or something. I mean, does it increase? Are you expecting that there's going to be common variation in at least some of these rare variant genes? I mean, it seems that would be the likely conclusion, and maybe it's just you don't have enough power in your...
There's definitely a power issue. We have one example, but the other 102 genes, there wasn't much there, but power is clearly a major issue. All right, so maybe I go on really quickly. So the idea of precision medicine is, you know, in terms of the rare variant spaces, you stratify people based on their gene mutation and develop a treatment for them using that pathway of, you know, stem cells, animals, and so on. So I already told you that, you know, no matter how you slice it, autism is heterogeneous. It's not excitatory cells, it's not synaptic genes, there are many ways to autism. So heterogeneity is a very, very, very confounding issue in terms of drug discovery and development. One thing that's good, if you will, is that most of the mutations are that we know of today are dominant loss of function mutations. Why is that good? There's a second allele, and if you could figure out how to get that other allele to express more, you have a platform for treatments in autism. One thing I mentioned in passing, which is a complexity, is that it's a U-shaped curve, right? So remember I told you trisomy has a phenotype, but not so severe. Monosomy has a phenotype more severe, and disomy is typical. But we already knew from these recurrent CMVs I talked about earlier, where they're both, or by nature of the way the genome is organized, you can get both deletions and duplications. We already knew that for pretty much every one of those intervals, there is a syndrome associated with both the deletion and the duplication. I've given you some examples on the left. So guess what? You know, we all know, we know very well that the stoichiometry of proteins in the synapse is very, very, very tightly regulated. There are these huge multimeric complexes. Too much of any one part of that complex is going to disrupt the complex, but too little does the same thing. Sorry, was there a question? Yeah. So the complexity here is that, first of all, it reminds you that it's not just putting more, sometimes it's putting less, and you have to worry about overshooting, or you may need to worry about overshooting. The important thing also to remember, and I highlighted this earlier, is that the disorders we're talking about, it's not just autism. These are very severe disorders. They really are of a severity that, you know, you probably want to take very significant steps to try and ameliorate the condition. And as there are many studies for many of these genes that show if you turn the gene back on later in development and even in adulthood, as in this paper, you get recovery. So SHANK3 is down, then you turn it back up, and you get behavioral recovery. I will say there was a caveat with the tamoxifen approach, and then there's been another paper that says it's all okay. This is certainly true, so good news. But basically, we're pretty sure from preclinical studies that you can turn the gene back on, you know, later than in utero and get some recovery. Okay, and as I said before, they're also relatively common. So they meet orphan designation, but they're not so vanishingly small that there's no path towards financial, you know, success. Okay, and what I've also emphasized a few times is that the effect sizes are huge, right? I said top genes, minimum of 20-fold increase in risk. Some of them are probably close to Mendelian, and even the transmitted loss of function have an odds ratio of 5. Okay, so there's not a single pathway, which is bad. They're overwhelmingly dominant, which is good. They're loss of function, which, you know, maybe we can get the other allele manipulated. They have a U-shaped curve, which is complicated. There is a huge unmet need, and as I mentioned, in some studies in rodents, you turn the gene back on for some of these genes, you get improvements. They're fairly common, they're highly penetrant. I don't see, looking at bullet point 1, that you know, the etiological heterogeneity is so high, it's hard to imagine how to avoid doing a precision medicine approach in a disorder like this. Okay, so remember this little slide I showed you about Miseryoff syndrome? And everybody in the audience said they would definitely jump on that right away. So I said, okay, there's not one Miseryoff syndrome. There are many Miseryoff syndromes. Miseryoff is a word in Hebrew which means fake. So, you know, it was really quite impressive, and in the end, it generated a lot of conversation and also follow-up, including from Takeda. They all said unambiguously, with 100% certitude, that if I could show them a gene that had all these properties, remember all these properties, they would be working on it. So, you know, it's clear that SHANK3 is the cause of Phelan-McDermott syndrome. The test is easy. You sequence the gene, high conversion. We don't see a single control in Nomad that has a mutation in this gene that has typical development. Construct validity is high. I showed you the animal model where you turn it back on, things get better. There's another paper that I also mentioned. Prevalence is about that. The Family Foundation there's now 2 family foundations. They're super engaged. Seaver Center Alex Calypso has created a network of sites across the country that do phenotyping on the syndrome. So we have like clinical trial ready sites all across the country. FDR is clearly very supportive in conversations about all these orphan disorders. And you know, if somebody got an indication for a drug for SHANK3, I bet we try it in SHANK2. I bet we try it in other synaptic forms of autism, right? So SHANK3 is just one example. I could give you the same list for 10 other genes. And so when I kind of did the big reveal, which was there are lots of genes that meet these criteria, but for some reason, biotech and pharma has not really gotten on board with that. They've gotten on board with Fragile X and with Rett syndrome. And now, really since that time, there are a bunch of companies working on SHANK3, and we're trying to kind of get the word out on the other 20 some odd genes that we think clearly already meet all these criteria. And so we do think that there is a path for precision medicine. We don't really understand why. I mean, SHANK3, when we were working on it 10 years ago, it wasn't by accident. We already knew that it was a significant gene. So imagine if you were the company that started with SHANK3 10 years ago, you would pretty much dominate the market, right? So I'm going to stop there. This is the Seaver Center and their advisors and their external site visit team. But I want to thank everybody at the Seaver Center. It really is a wonderful, you know, I love coming to work every day because I can learn about many, many, many different things across, you know, from molecular to cellular to animals to patients. And we all like to kind of work together and kind of, you know, meet in big groups. So I'll stop there.
Joe, I'm just wondering, if I bring any child to you who has autism, I say to you, he has autism, what are the chances that he would have in his genes some combination of these rare mutations? So in other words, like it's rare in autism, the mutations are rare in autism, but what about the combination of genes? Are they together still very rare?
So we think that if you take all of our genes, about 5%-ish of all cases with autism will have a mutation in one of those genes. If you include the copy number variants, the deletions and duplications that we know are increased risk, it's probably closer to 20%. So you can come up with a genetic etiology in about 20% of autism today.
Thank you.
Well, thank you for your attention.
Joseph, thanks very much for a great talk.